Hi, good morning. I have. Yeah. Okay. So, good morning to everyone. First of all, thank you very much, Osvaldo, to this organization of ECODIM, Matías, Monse, Marcela, for inviting me to give a lecture. For me, it's really a pleasure and an honor to be here in this stage, having you all of here presentially. It has been a long time since we don't met for this amazing course, so it's very a pleasure, and I thank you, Osvaldo, for inviting me to this symposium. I hope it's okay. See, okay. So I am not sure if this works. Okay. The title of my lecture, of my talk for today is, did we have? I will talk first about some Antarctic facts. I will give a kind of introduction about why we are talking about sponges as primitive gut in Antarctica. Then I will go through functional potential of microorganisms associated with Antarctic sponge, host specificity and acquisition mode in Antarctic sponge microbiomes. I will talk about some main guilds that we identify in the sponges, and I hope that you can take some final message so we can talk after this about the interesting sponge models for symbiosis. First of all, I don't know if you know, but Antarctica is an extreme environment. So I will try to convince you why an extreme environments involve an extreme association in particular conditions. Antarctica is the driest continent on the planet with more than 100 millimeters of rainfall per year on average. As an example, just in Santiago de Chile, we have rain for more than 300 millimeters a year. So imagine all the Antarctic continent with rainfall at just 100 millimeters per year. But it's also the place where the lowest temperature has been recorded. This one was almost minus 90 Celsius degrees, with a seawater temperature between 1.8 and 3.5 Celsius degrees. And is also the windiest continent on Earth, with winds with more than 300 kilometers per hour. Antarctica is also our main reservoir for fresh water. In Antarctica, it's okay, no, are my lens. With Antarctica contains 85% of all the water that we have, the fresh waters that we have for Earth. This is very important for us because this is our main reservoir. Here is where the most glaciers are available. So in the rest of the world, we have just 15%, and all of other reservoirs of fresh waters is in this continent. But Antarctica also helps to control the climate of the planet. While all the world's ocean reflects only a 10% of all the radiation that impact the Earth, the Antarctic continent can reflect more than 90% of this radiation, so it controls and can avoid that all the heat that entered the planet can be retained, so we reflect to the space. This important phenomenon helps regulate all the temperatures and prevent the planet from absorbing a lot of heat or radiation, which can be trapped in the atmosphere due to greenhouse gases. So with all of this fact, one of our questions, our very early questions in microbial ecology for polar environments was on whom the iconic Antarctic fauna depends. And here I am showing you this iconic fauna, you know, whales, penguins, sea mammals, all the animals that we recognize as typical from Antarctica, they relies on microorganisms. In most ecosystems where terrestrial can be supported, the primary productivity can be supported by plants, in Antarctica, thanks to the phytoplankton and to the contribution of this enormous amount of solar radiation, we have that the phytoplankton is the main responsible of the whole primary productivity in this continent. And here I show you an image of the ocean chlorophyll. So you can see, I don't know if this works, you can see this part where you see this red color here, the amount of chlorophyll that surrounded the Antarctic continent, and this fueled all the ecosystem through the higher trophic level. But one of the other extreme characteristic of this continent is that this can be only happen during summer. So another extreme condition of Antarctica is in fact the extreme seasonality. And this means that during the summer period, we have a lot of energy entering into the system. We had a lot of sun, a lot of solar radiation, a lot of phytoplankton, a lot of productivity, a lot of food for the ecosystem. But during winter, as you can see, there is almost no primary productivity, no chlorophyll, and most of this part is covered by ice. So in the Southern Ocean and the water that surrounds Antarctica, the connections between members of the microbial food and the microbial loop is very important. And most of all, because this makes that the coupling between the benthic and the pelagic ecosystem can be really tight. Here I show you that this coupling of benthic and pelagic ecosystem functions through a range of biological and physical processes that plays, of course, a key role in the biogeochemistry and in the ecological function of the complete Southern Ocean. Mainly because of the, I don't know if you see when I, yeah. Mainly because of melting of sea ice and a large input of iron. Through this melting of ice, phytoplankton blooms increased during summer, and this also fueled all the rest of the structure that can transport this nutrient influx to the rest of the ecosystem. In the part of this system, of course, we have the microbial loop, and in this case, processes driving carbon transfer to higher trophic level and flux to the deep ocean are mediated, of course, mainly by microorganisms. This food web includes the microbial loop in which dissolved carbon substrates fuel the growth of bacteria and archaea, which are subsequently consumed by protists, grazers, and all other members of the food web, canalizing the transfer of energy and carbon to the other levels. So, and probably you know that also Antarctica is one of the places that is most experiencing the effects of climate change. We already know that the temperature around Antarctica, and mainly in the western part of the peninsula, is being increasingly warm in the last 20 years. You can see here the amount of increase of sea temperatures in this area that also correlates with a big mass, a big loss of ice melting and a big mass of less ice in this part of the continent. In this part, I will change a little bit through the Antarctic facts to explaining you why we consider sponges as primitive cats. One of the interesting characteristics of this extreme environment is that we have a lot of benthic coverage for one kind of organisms that are marine sponges. Marine sponges can occupy in the Antarctic environment almost 80% of the substrate. Almost all the benthic substrates are occupied by different kinds of sponge species in these environments, and they, of course, play a critical role in the coupling of benthic and pelagic system through the filter feeding capabilities that this kind of organism has. Sponges are incredibly diverse in color, in shape, are a group of animals appear many years ago with more than 6,000 taxonomically described species, and that, as I say, abundant in all benthic habitats. These kind of animals are also very rich in bioactive compounds. In fact, it's one of the main sources of biotechnological products made by marine organisms, with more than 7,000 described bioactive compounds that are until now very useful for the pharmaceutical companies. But sponges are also the most ancient animals that appear in the evolution. These very simple organisms appear 600 million of years ago, and because of that are the oldest known extant animal microbe symbiosis. And this is why we talk about that this is our model for a primitive cat, and we are trying to understand many molecular bases of this interaction using this kind of organisms as a model study to study microorganisms. And why, you think? Well, that is because this organism and most of the capabilities of structuring benthic ecosystem and coupling pelagic and benthic processes is mediated by microorganisms. Sponges host an incredible large number of bacteria, archaea, and microbial eukaryotes that lives in a symbiotic relationship within the sponges. These processes of symbiosis represent our most basal understanding of the symbiosis interaction. Here I show you that based on a cellular grade model that are sponges, we can identify that there are the basal place of the phylogeny of animals. They represent the most simplest way to understand interaction between microorganisms and the complexity of animals that at this stage just appear with sponges. So this is our principle of study, that we are trying to understand the molecular basis into symbiotic processes between host, and mostly animal host, and microorganisms. And this is because microorganisms can be up to the 60% of the whole biomass of the sponge. That means that it's a microorganism bag, mainly, with some processes, but just very high amount of bacteria, archaea, and eukaryotes. And this is because of the filter feeding capabilities and mode of nutrition that sponges has. They filter seawater through this channel system, and they, by process that we don't understand really why, sponges can select which microorganism they will retain as symbiotic partners, and which will be used as food to enter into the energy and carbon fluxes from the sponges. Sponges also are very important in a process that now we call a sponge loop, in which dissolved organic matter can be transferred from lower trophic level to higher trophic level based on these capabilities of filtering of large amount of carbon and large amount of seawater. So the sponge loop is an alternative pathway for transferring them to higher trophic level, and even if was originally identified in tropical ecosystem, recently we identified that this sponge loop can also occur in cold water. And in fact, in cold water, the transfer of this amount of carbon that sponges can filter can be even higher than the amount of carbon that can be fueled and circled by the microbial loop. Microbial symbionts from marine sponges have also complex metabolic repertoires and mediates very important nutrients and energy transfer mechanism. We have identified, based on different approaches, that in sponges are occurring different kind of functional processes, photosynthesis, methane oxidation, sulfate reduction, dehalogenation, but most importantly is nitrogen, that we have identified many or all the steps from the nitrogen cycles occurring inside the sponges, and also a process related to phosphorus sequestration. Here I show you evidence of the different steps in the nitrogen cycles that we have identified in sponges: nitrification, denitrification, ammonification, ammonia oxidation, and everything. And also, we identified that inside the sponges, there are important metabolism related to phosphorus sequestration in the form of granules of polyphosphates that can be used during different steps or different stage of the sponges, because during the filter feeding, the process of entering seawater mediate steps of oxygenation inside the sponges and low oxygen. So during this process, sponges can accumulate, mainly the bacteria inside sponges can accumulate polyphosphate to retain energy that can be released after that as a mode of interaction with the host. But also, sponges are a rich reservoir of diversity, of microbial diversity. Here I show you a heat map that shows the taxonomic profile of a big global study trying to understand the microbial composition of the whole communities of different sponges. However, this not include polar sponges. Be careful about that. The important thing here is that in this study, authors identify all the known phyla that are in bacteria are in fact can be found inside sponges. So the amount of diversity is gigantic. The other thing is that the diversity inside sponge is so large that it can be even higher than the diversity that we can find in seawater. And here I show you this summarize of this global study in which sponges rarefaction, as you can see, is much higher than the amount of diversity that we can obtain for seawater. Microbial symbionts from marine sponges are also very diverse, as I told you, but there are some insights that we can identify that there are also host-specific and different from the surrounding seawater. Here you can show that most of the sponge bacterial members are members of the Proteobacteria, Actinobacteria. In tropical and temperate system, Cyanobacteria are also very important. And here you can see that if we compare different kind of sponge species, you can identify that different sponge species has a different compositional pattern. and that is also completely different with the community composition that we found in the seawater. And here you can see this is the seawater, and these are different kind of sponge species, and you can recognize that this is a cluster and this is other one. So we have this differentiation that I told you before. So in some way, the sponge can select or decide which microorganisms they will retain and which will be used as food. The problem is most of the studies, let's say, all of the studies that I told you here came from tropical or temperate environments. So even if we know a lot of the sponge symbiosis for very beautiful environments, for the extreme and cold and wildest environment from Antarctica, we don't know so much. Our first analysis started with the taxonomic signatures. We tried to understand who are the microorganisms living inside Antarctic sponges. And our first study was based on 8 sponge species with samples taken from the Fildes Bay here in the Western Antarctic Peninsula. We performed for the first time a metagenomic approach trying to understand the community composition of the microbial community from these Antarctic sponges. And normally, symbiosis focus between animals and many other, and in many models, try to focus on bacteria, and we try here to keep the understanding, not just, some disconfiguration. Yeah, but no problem. We try to understand not just bacteria archaea, that is this plot, but also microbial. No, it's completely lost because it's the same. But trust me that this is completely different. This is for bacteria, and if you can see the relation between richness and diversity is completely different for eukaryotes. So we recognize so that in the bacteria archaea part of the microbial community, most of our 8 sponge species have different microbial communities. Most of them belong to proteobacteria, but also verrucomicrobia, actinobacteria, bacteroidetes, and also an important contribution here, please, of Taumarchiota in the other taxonomy assignment. And in the case of eukarya, we detect a lot of photosynthetic groups like stremonophiles, but also a big contribution of dinoflagellates from Syndyniales, that is mainly a group of parasites. So based on that we can understand that 3-domain microbial community from Antarctic sponges are, as we see for other environments also, are remarkably diverse. About the host specificity, we could not say so much because here we have only one individual per sponge species, several samples, but just one individual. So we keep these questions a little bit in asterisk, and absolutely different from the seawater, from the surrounding seawater, that is for the bacteria and for the eukarya component. When we try to compare the taxonomic signatures from our Antarctic sponge against other environment sponge, we find an interesting results, but also an impediment to continue. There was no eukaryote studies to compare really our microbial eukaryotes analysis based on taxonomic, on Antarctic sponges. But for bacteria, here you can see the squares are polar sponges, and you can see that they are completely different. So we detect differences in the composition of the sponge-associated bacteria. between Antarctic and tropical environment. This is mainly because of the presence of the absence of cyanobacteria in our Antarctic sponges. As you can see here, there is no evidence of cyanobacteria, and this is a very important difference between temporal, tropical, and Antarctic or polar sponges. We move to the functional potential of microorganisms associated to these sponges. We try with some metagenomic approaches, trying to understand not just one single gene for taxonomic approaches, but also the whole metabolic potential using metagenomic sequencing. We use 6 different metagenomes. This was one of the first studies from my first students, also my first PhD student that was also a student from Ecodim. So we are recognized that it's a kind of family. Somebody asked me who are the descendants of the speakers or all the former students that are here. It's very nice. So this first study was very interesting, but then we find that the amount of annotated sequence was very low. Only 20% or only 50% of our first approaches to do metagenomic were unassigned. So we recognize that we are working with an environment that has very, very small contribution in the whole data sets, in the whole databases. And this is a complication. But we focus in those metagenomes that we have more amount of data to analyze. And first of all, we recognize that functions related to symbiotic lifestyles are increased in the microbiomes, in the sponge microbiomes compared to the seawater. What I show you are CRISPR-Cas system, toxin-antitoxin, restriction modification system, different kinds of mobile genetic elements, and eukaryotic-like proteins. All of these elements, all of these features are molecular elements that mediated the symbiosis between microbes and the host. And we identified that all of these processes were absolutely enriched in the sponge compared to the seawater. We also identify different modes of biogeochemistry cycling, mostly related to nitrogen. We identify ammonia oxidation, nitrate, and nitrate oxidation also. And we identify that most of these processes were performed, perhaps, by Nitrosopumilus, and here I show you a synteny showing what we understand from our Nitrosopumilus putative candidate to perform these steps with the one that has the genome already sequenced and are in the database. Most important, we identify that we can find 4 of the 6 described autotrophic carbon fixation pathway that are already described. This was the most extensive autotrophic carbon fixation repertoire found in sponges until now, because we were able to find that in Antarctic sponges, potentially, because these are metagenomic data, are occurring the Calvin cycle, the reductive TCA, so tricarboxylic acid cycle, the 3-hydroxypropionate, 3-hydroxybutyrate cycle, and also the Wundt-Lundial. So a very broad spectrum of ways to fix carbon. Please remember that I told you that one of the main characteristics of the extreme environment from Antarctica is the seasonal variability. So we have a lot of carbon input of energy and primary production during summer, but during winter, what happened with all the ecosystem? Perhaps this could be a way to understand how the sponge fuel the nutritional requirements with other kind of autotrophic carbon fixation mediated by bacteria. We move a little bit and we try to understand also what happens with isolated bacteria that we could culture from the sponge, from the Antarctic sponge. This was not an easy task because, as you can understand, symbiosis imply a kind of molecular and metabolic interaction, and the culture of symbionts is normally a hard task, but we could understand the genomes and identify the genomes from several strains that were isolated from Iophon, one of our sponge species, and dendrilla. The species, the bacteria species that we could culture at the lab were associated with Sporosarcina, Cellulophaga, and Nesteronconia, and we identified that Sporosarcina and Nesteronconia were most probably related to novel species that are undescribed in the databases. We also try to understand why these sponge symbionts could be or not could be cultured in the lab. And we detect that this kind of species that we have isolated live or thrive almost indistinctly between sponges and seawater. So we could find these same species in similar amount in other environment, sponges from other environment, seawater microbiomes, and also in Antarctic sponge microbiomes, the one that we were studying here. So cultivated sponges inhabit similarly marine sponges and seawater. We wanted to focus a little bit more in the host specificity. I told you that for other environments, the signatures for host specificity was almost clear, but at least for our perspective, for the Antarctic sponges, this was not so clear. This was our first understanding of host specificity, and because of the small number of samples of replicates that we could obtain at that moment, the results were not so clear. So we increased the replicates and we moved to another geographical location from the Western Antarctic Peninsula, and we focus on 3 sponge species, Aliclona, Hymeniacidium torquata, and Isiodictyva querulensis, but using only 3 species, but a big, no, not big, let's come on, a higher number of replicates, trying to understand if the taxonomic signatures were really conserved between or within the sponges or intra-species. And we see that similar bacterial community composition were present in these different sponge species, but among the same sponge species, we can see that these samples are more similar to this one and to this one. And that Hymeniacidium torquata showed the highest level of intraspecificity. So we try to understand if this symbiotic relationship was equally conserved between these 3 sponge species, and we recognize that some of them were mostly important based on, for example, cytophagales, while others were related to other sponge species, and that the force of the interaction between the microbial partners and the sponges was different based on the sponge species. And that also we can find a low number of species-specific otus with important difference between the total abundance accounted for these otus. And here, for example, you can see that there is 60%, 20%, or 45% of these accounts for the participation of the core otu in the whole microbial community. We move a little bit to try to understand more functional and more community composition in a deeper way. So here we combine not 16S or 18S tag sequencing, so our typical taxonomic approach, with our metagenomic approach that could be produce metagenomes, assembled genomes, or MAGs, so we can try to understand which were the main guilds in the Antarctic sponge olobionts and how we can connect the ecosystem functioning that is happening in Antarctica with what is happening in the sponge as an holobiont. And here we increase or we combine all the samples that we have at that time that were 26 sponge samples, 19 sponge species that correspond to 2 classes, 6 orders, and 12 families. And using a bigger data sets, we start to recognize different patterns because our picture, our view of the microbiome from this Antarctic sponge start to be a little bit more clear. And here I show you that, for example, for bacteria, archaea, and for eukarya, the shared and exclusive ASVs, our proxy to species, from the microbial community of sponges and seawater was completely different. Please see that here, the shared proxy of species, ASVs, were 10% for bacteria archaea, with a specific or exclusive community that correspond to almost 80% of the total community composition, but in eukarya, the picture was completely different, and the amount of sharing was much, much more, almost 70%, with just a 50% of specific or exclusive species that we can find in the sponges. We ask also for co-occurrence patterns, and we identified that the most important groups that were co-occurring within the Antarctic sponge community in terms of bacteria were alpha, verrucomicrobia, flavobacteria, and gamma proteobacteria. These are the genus or genera that were most connected with most important interactions, putative interaction with the whole community. We infer also the taxonomic composition not using 16S marker genes, so using the whole metagenomic datasets, and we identified that, as previously seen, in our Antarctic sponges, the microbial community composition was dominated by gamma, alpha, proteobacteria, planktomycetes. If you can see here, there is not so many groups that are the most abundant. It was completely in agreement with our 16S previous results. So for bacteria, the metagenomic datasets and the 16S completely agrees. This is because most of the databases are enriched in ribosomal genes, for bacteria, but when we talk about eukarya, we see a completely different picture. The microbial eukaryotes associated with sponge were characterized by a presence of a much diverse community, but with very low abundant members. And in this case, we could not find an important correlation or agreement with our 18S sequencing. Here, for example, appear a lot of fungi sequences that we were not detecting in the 18S database. So there is still a gap of knowledge, a big challenge trying to combine these two approaches. We see specifically what happened with the biogeochemical cycling in the Antarctic sponge holobiont. And here I show you 3 circus plots in which you can see the contribution of our different Antarctic sponges and different key markers that represent carbon. For example, here we detect RBCL, RBCS. For nitrogen, a lot of prevalence of genes related to nitrogen cycling and also for sulfur. So we detect these main microbial guilds related to carbon fixation, mainly based on gamma proteobacteria, but also for sequence related to diatoms, phaeocystis, and primnesiales in the eukaryote parts. In the case of the nitrogen cycling, we identified that this guild was mainly represented by nitrospina, and in the case of the sulfur cycling, we recognized that there was a lot of members that could have these sulfur cycling capabilities or metabolic potential. We also recognized that, we don't recognize, we were able to assemble 28 metagenomes assembled from genomes, and we detect a high level of novelty. Max shared only between 76 and 95% of average nucleotide identity, so our proxy to species in the genomes, compared with all the database, mainly with the GTDB, that is at least now the main database for bacteria. So that means that all of them correspond, the 28 max could potentially correspond to novel bacterial genomes. We also identify that in this genome there was a strong signal of streamlining. For example, here I show you the genome size, GC content, and coding density, and we can see that, for example, the Lovibriona, a member of one of the most important members of the bacterial community here in this sponge species, has a very reduced genome compared with the counterpart from free living. Oh, no se ve tan bien. Yeah, no importa. We identified that several beans, several max, first of all, being 20 that correspond to Nitrosopumilus, and where we can detect an autotrophic carbon fixation based on this 3HP, 3HB cycle, ammonia transport, nitrate oxidation, nitrite reduction. So we could identify that being 20 correspond to an ammonia oxidation, oxidizing archaea. In the case, you don't see, but here is Nitrosopumilus. Being 24, corresponding to Nitrosomonadase, that belongs to mainly to the ammonia oxidizing bacteria. We find here the CBB cycles, also, of course, ammonia oxidation, nitric oxide reduction. Being 07, belonging to gamma proteobacteria, that could be assigned to a sulfur oxidizing bacteria, being 03 to a nitrate oxidizing bacteria, and being 28 that was very similar to a sponge symbiont from a sponge from a tropical environment that is also a sulfur oxidizing bacteria. And we also know or show that there was a prevalence of archaea that are capable of oxidizing ammonia against bacteria that are oxidizers of ammonia. We also detect a lot of symbiotic genes, but here you can see the amount of carbon fixation pathway, nitrogen fixation pathway, sulfur, phosphorus. But what is very interesting that no se ve tan bien. What is very interesting that we can find a lot of cold adaptation features in these genomes, and of course symbiotic genes, the same as I told you previously, so CRISPR-Cas, toxin antitoxin restriction, modification system, all of the molecular mechanisms that we know that mediate the symbiosis between microbes and the sponges. We also identify a lot of genes related to metabolisms to carbohydrate. This was very logical, but not previously reported for Antarctic sponge. So was very nice to find a big amount of Casey enzymes or carbohydrate active enzymes, all the metabolisms related to carbohydrate. Imagine that the microbes that are filtered by the sponge need to be located physically inside the sponge tissue. And for this, you need metabolic activities related to degradation or restructuring of carbohydrates. So here I show you the most important beans, but it's difficult to see, but it's bean 07, bean 20, bean 20 is here. That is related to glycoside transferase and with glycoside hydrolase that were very, very important in our Antarctic sponges. So with all this kind of evidence, we now think that we have a deeper understanding of the Antarctic sponges. We are recognizing that some of the main features that we know for other sponges, for other environments, are in fact represented also in polar sponge holobionts. But other characteristics are very different compared to the tropical or temperate environment. I am okay in time. So, as a summary, Antarctic sponge microbiomes have a particular composition and a very different functional signatures compared to the other sponge from other environments. The unique metabolic potential of Antarctic sponge microbiomes, we think that could help the holobiont, this special Antarctic holobiont, to survive in this Antarctic system. We identified that the genomes of sponge associated strains are different from the related genomes from the free-living relatives due to the symbiotic characteristics and some extreme condition of Antarctica. We recognize that the relationship between the Antarctic sponge microbiome and their host was not equally conserved between the sponge species, with important difference in the acquisition of the sponge microbiome among different host species. We showed that bacteria have a stronger association with the host compared to eukarya. We don't understand until now why. We saw these clear patterns or these different patterns between bacteria, archaea, and eukarya, but with the time, this differentiation get more clear, so they are not comparable. And finally, that we could find specific genomic features and functional traits of the more abundant microbial members of the Antarctic sponge holobiont. So it's more easily to understand which functions are in fact performed by the sponge holobiont in this system. So finally, perhaps my main message is that these extreme environments implies an extreme association, and that we could really understand these sponges as model for primitive guts. I want to thank finally all the team. Patricio is also here, but here are also most of this team are former ECODIM students. So this is a very important place to say thanks to all the members at the lab for all the work, and also to the funding that we have available at the lab, Fondesit, the INACH, Millennium Institute. We are part now of Millennium Nucleus and the Fondab Ideal. So thank you very much, and more than welcome to questions.
Well, Thank you, Nicole, about that. We have time for 2 or 3 questions. So please.
Well, thank you for your presentation. I think that it was very interesting one. Well, I have 2 questions, and I don't know if I can do both of these, but one of these is among the environmental factors, such as the chemical, physical, dissolved gases, or even the seasonality, the environmental seasonality, sorry, which could be the main factor that determine or affect the sponge's alluvium and also may affect the potential metabolisms or of these entire communities?
That is a huge question. It's like we can talk for hours. I think what we are trying to understand is like the coupling between the holobiont as benthic pelagic couplers with what is happening in the surrounding system. You know, here I do not show all the chemical or the physical drivers, but particularly in the place where we are studying, so the Western Antarctic Peninsula is a place where we can find compared to other parts of Antarctica, a lot of anthropogenic stressors also. So that is another kind of interaction that we are not considering here because we are just seeing what is happening in a particular place of the peninsula. So I think that there are many connections, many of them that we do not understand so clear until now. Perhaps that is my main answer.
Yeah, it's quite difficult. And I don't know if I can do the other. Well, I think that these cyanobacteria approaches, it's quite interesting. So as I know, cyanobacteria are the most ancient microorganisms on Earth. And in this context, how can we explain the absence of these microorganisms and the develop of the sponges on a evolutionary point of view?
Yeah, this is a very interesting question. Thank you very much. Because the reason why we think that cyanobacteria are absent in Antarctic sponges is because they are not present in cold waters. Normally, cyanobacteria have a distribution that decreases from polar to poles. So we do not have so much cyanobacteria in the seawater surrounding. So in a way, this was like, of course, this was obvious, you know, but in any case, was very interesting to separate completely the community and could understand why they are so different. And we think that the autotrophic role that perform cyanobacteria with the sponges in tropical and coral, in tropical and temperate environments are replaced by autotrophic bacteria that lives in the Antarctic sponge and that could supply not just during summer, that then you have this amount of diatoms, dinoflagellates, and other microalgae during summer, phytoplankton, but during winter.
There's another question?
Hi, Nicole. Thank you very much for your presentation. I was really curious about this reduction of the genome, like, I don't know how to say, size. Because we know that genome reduction, it's like a form of reducing energy like consumption. So how is this related to the specificity of different species of sponges? Are you also studying like the sponge genome and how the interaction? Because if they're reducing this size, are they losing some functions, and how does the interactions between the other components in the microbiome change? Is that like an approach you're taking?
Thank you very much, Belen. This was very interesting because I show you that for some max some genomes that we could assemble from the sponge microbiome, we detect these signatures of streamlining, so the genome get reduced. However, in other ones, we detect the opposite. And there is, most of the literature say that during symbiotic processes, you see this kind of reduction of the genome. But in some cases, you also detect this kind of expansion. And that is very interesting because that could mean that in some way, some bacteria are decreasing, so are reducing and streamlining, are getting more efficient, but other ones are not. That means that the environment is not really an environment that is like forcing the symbiotic relation, and that could in some way be, or try to understand, like a more like a free community, a seawater community than a host specific community. I don't know if that answered the question.
Is it on? Okay, great. Thanks, Nicole, for a great talk. Your hypothesis that that chemoautotrophy drives the association in winter is intriguing because I am trying to figure out what you think that electron donors, the energy sources are. And for the ammonium oxidizers, the sponge could be producing the ammonium, but then you've got a, what is the sponge taking? The sponge has to produce the ammonium. So that's a, what is it? Mobile perpetuum or perpetuum mobile. I always get it confused which way around it is. So that doesn't work. You're going to need electron donors from the environment. And I'm intrigued in what you think they could be.
Yeah, exactly. We think that based on the data that we have, we can recognize that ammonia can be released during the metabolic processes of the sponges, and that could fuel a little bit of this. And during winter, the thing is that we do not have we still have the proper amount of electron and elements to fulfill. And the growth of the sponges is still is low during winter. Overall is low, but still can be capable of return elements to the microbiomes. So I think, or we think, that this all is based on the benthic activity of the sponges. And one of the problems is that until now, and I don't know if we will be able to do that, we are not able to sample sponges during winter in Antarctica. So perhaps that is the main problem to really catch this part.
But you can sample them before the winter and after the winter.
Yeah, that is
right?
what.
And so, in theory, what you should see as organic material disappears from the seawaters, what you should see is either the metabolism goes down to the degree where you could postulate that the symbionts are becoming parasitic because I'm yeah, exactly, that they are eating up the sponge, and then as photosynthesis ramps up again in the spring. So it would be fascinating to, at least in the months before the winter and after the winter, to see if that cycle really happens.
Yeah, that is exactly because we could not go in winter. That was exactly our last sampling strategy, trying to touch what was happening at the beginning of the summer, at the beginning of the spring. That was our wishing list, but was almost not possible. But at the beginning of the spring and at the end of the summer or the beginning of the autumn. Yeah, that could be another option. Yeah, but that is exactly the idea, because until now, it's an interesting hypothesis, but this is only metagenomes. We are not looking until now. We have some transcriptome weighting, and we also have some genomes from the sponges that we are analyzing now. So just now we can see if there is more than hypothesis, and it's something that we can try to go and understand which are the equivalents for the energy transfer.
Hello. I have two questions, and I think that were simple. One of these, if you don't have much information about that, I request if you can tell me hypothetically. One, if the ambiental parameters, like the stationality of the ambient in this Antarctic zone, can change the microbes of the that are in symbiosis with the sponges, and if it is, they can have periodically symbiosis between phase and phase. And the other question I want to say, if the microbes that are in symbiosis with the sponges are can be transpassed to the descendants, or are only microbes that are around in the seawater?
Great, thank you. Thank you. So for the first question, next year I could answer you about the seasonality and how this changed during a time period that we have now 4 years, something like that. So we could really understand a little bit more of the dynamics, because until now I show you just like single pictures that could be very interesting, but perhaps do not reflect the proper dynamics or functionality. So I don't know. For the second question, these are not our experiments, but we know that part of the microbial community from the sponge can be vertically transferred, and most of them, but also can be acquired from the surrounding seawater. So it's a mixture. And it's not clear the proportion of these two factors, how impact the whole community, in which time can be more receptive to receive from the seawater, or less restrictive or less permissive.
Thank you all. We have time for one more? Yeah? Yeah, okay.
Yeah, I know that all of us want to go to the coffee, but I have two questions. One is from the point of view of the sponge. Can the sponge distinguish between microbial food and microbial partners? And the other one is more specific, is about the enzymes that you distinguish in your CASI. Do you have, okay, you have a lot of enzymes that are involved in the metabolism, but do you have there some glycosyl transferase, for example, to make EPS to colonize the sponge?
The first one.
The first one is
the recognition. Yeah. There is a lot of evidence that indirectly show that there are mechanisms of recognition based on the molecular mechanism that I show you, the presence of CRISPR, eukaryotic-like protein, toxin, antitoxin. So these mechanisms and the proportion of microbes inside the sponges, and that they have a completely different community compared to the seawater, suggest that there should be a mechanism to differentiate which microorganisms they will persist in the sponge tissue, and which will be feeded or converted into detritus and get into the food system. Yeah, so there is evidence. And in our case, it's very interesting because the diversity, the plethora of different mechanisms that we found for symbiotic association is very, very broad. And the second question?
About the glycosyltransferases.
Exactly. We found some of them related to exopolysaccharide. So we think also that these kind of mechanisms are perhaps in the middle or in a degree of relationship with the sponges. I mean that at some point these mechanisms help the microbiomes or the microbial partners to get in contact with the host as a beneficial partners. But in other way, the same mechanisms are the ones that are involved in the pathological or in the pathogenic association that the microbes has with the host. So this degree of different results that you can obtain from the same mechanism is very interesting, but that means that at some point this could be a dynamic flow between beneficial and perhaps commensalist and then pathogenic bacteria or other microbes. So like there are not always good and not always bad, something like that.
Okay, thank you very much. Thank you, Nicole.